Hi all. Today we will explore different laboratory methods used to identify biological molecules. Okay, so this toolkit focuses on detecting sugars, proteins, lipids, and starch in unknown food samples. So these tests are commonly used in biology laboratories and help scientists analyze the chemical composition of substances. Okay, so let's see how to do it. So there are four biological targets. What are they? Before testing, we first identify the four major biological molecules that we are looking for. So that are starch, proteins, lipids, and sugars. Okay, so each molecule react differently with the specific chemicals. By observing these reactions, we can determine which molecules are present in the sample. Okay, so let's see the testing for starch. How will you identify whether starch is present in some food or not? So to test for starch, we use iodine solution. So which solution we need? Iodine. Only few drops are needed, like 2 to 3 drops are needed. Okay. And no heating is required. If starch is present, iodine changes from orange brown to blue black. Okay. So if there is no starch, the solution remains orange brown. So if there is no starch, it will remain orange brown. If there is starch, the orange brown will change to blue black. Right. So this is a quick and reliable starch detection method, right? So the uh, advantage is that we don't need to heat the particles. Okay. Second test is for proteins. Let's see how to test protein. So proteins are detected using Biuret test. Okay. What is Biuret test? So equal amounts of the sample and Biuret solution. So what we need here, the sample and the Biuret solution are mixed together and no heating is required. Here also we don't need heat. So a negative result stays blue. So if there is no protein, the solution remains blue. While a positive result changes from blue to purple. So if proteins are present, it will change from blue to purple. Okay. So the purple color confirms that the presence of protein. So the solution or the product has proteins. Got it. So always keep in mind we need equal amounts, not a few drops. Okay. Now let's check for lipids. How to identify whether there is lipids or not. So basically, lipids are identified using the emulsion test. Okay. First, the sample is mixed with the ethanol. Okay, what we have to do? First, we have to mix it with the ethanol. Then add it to cold water. Okay, so we'll be adding to cold water. If lipids are present, a cloudy white emulsion forms. So there will be a milky white emulsion if there is lipids. If not, the solution remains clear. So if there is no lipids, the solution will remain clear. Got it? So the cloudy appearance happens because lipids separate out from the solution. So if there is no lipids, there won't be any color change, right? So lipids precipitate out of the solution, causing visible cloudy phase shift. So this is how it will be visible if there are lipids. If not, the solution will remain in a clear form. So what we need here, we need clear water, cold clear water required. Okay. Now let's see the testing for sugar. Okay. So the test name is called a Benedict test, right? So let's see how to do it. So reducing sugar are tested using Benedict solution. Okay. So the sample must be heated in a water bath above 80 degrees Celsius. So we have the Benedict solution and we'll be heating the water, right? Heating it in a water bath up to 80 degrees Celsius. Then, if reducing sugars are present, the solution changes color from blue to green, right? And what if there is no reducing sugar present? The color will remain the same, right? So if there is the reducing sugar is present, the solution changes from blue to green, yellow, orange, or brick red, depending on the concentration. Okay. So this is how the positive results will look like, and this is how the negative result will look like. Okay. So what we need here, we need a water bath to 80 degree. Okay. And how will you estimate the sugar concentration based on the colors? If there is reducing sugar present, we need to identify the concentration, right? So faster color change is equal to higher sugar concentration. So the Benedict test can also estimate sugar concentration. Faster and stronger color changes indicate higher sugar concentration. So scientists compare unknown samples with the standard reference solutions to estimate how much reducing sugar is present. Got it? So from an unknown sample, through this test, we can see different combinations. Okay, different percentages will be there. So blue means 0, uh, green means light, and yellow is more so concentrated. Orange means which is comparatively higher, and brick red is the highest concentration. Okay. So now let's see the testing for non-reducing sugars. How will you identify non-reducing sugars? So non-reducing sugars require two-stage process. Firstly, we need to treat it with the hydrochloric acid that is added, and the sample is heated to break complex sugars into simpler sugars. So first step is hydrolysis. We will add sample and hydrochloric acid, right? Then we'll heat it for 2 minutes, and the sugar will be completely broken, okay, into monosaccharides. Then what we have to do? Then sodium hydroxide is added to neutralize the acid. Okay. So second stage will be neutralization and detection, right? So from the input, right? We have an input here that is monosaccharides. So we will add sodium hydroxide into the mixture to make this alkaline, right? After that, a standard Benedict test is performed. So perform the standard Benedict test. Then we can confirm the color change. So a color change confirms that non-reducing sugars were originally present. Okay. So color changes confirm original presence of non-reducing sugars. Clear? So this is what we have learned so far. So for identifying sugar, if it is reducing, Benedict test, thermal protocol, we need a temperature up to 80 or greater than 80 degree. In a negative state or initial state will be blue. Positive indication will be the color codes, right? From blue to green, green to yellow, yellow to orange, and green based on the concentration. And for proteins, it is Biuret test, and we don't need any heat. Initially, it will be blue. It will stay the same if no proteins are present, but if proteins are present, it will turn to white or purple. And for lipids, we need ethanol and water. So we'll be using cold water. It will remain colorless. And if there is any presence of lipids, it will form a cloudy mixture, right? And for starch, iodine test, no heat is required. If there is no starch present, it will remain orange. If there are presence of starch, it will turn to blue, right? So these are the biochemical tests essential for essential tools in biology. Okay. And this will help the scientists understand the composition of food and other biological samples. Thank you for listening.